Mark asked me to talk about the future, but before I go to the future of immunotherapy, I must say thank you for a lot of people that made this immunotherapy possible. First of all is the AMBL, that's the COG study, those patients and the family that allows their children to participate, especially in the randomized trial. That is a very tough one because at the time there's no foresight whether it's going to work or not, but certainly it's a treatment with a lot of side effect. So I appreciate to their participation. And then there are lots of COG leadership involved. Many of the names I think are familiar to you. And then on the other side, the CTAP or the NCI in the USA, which made a big contribution in providing the phase 3 material, the antibody, to make it possible. And initially, all the phase 1, phase 2 are done at UC San Diego. It's a single institute. These are my postdoctor. Some of them are from this part of the world, from Europe. Most importantly, we have to thank Dr. Rolf Reisfeld, who's the inventor of the very original anti-GD2 antibody. So I know you heard a lot about antibodies, but what is antibody? So I thought I would start from very basic. So antibodies are molecules produced by our B cells. And our B cells usually have these so-called immune or antibody molecule on their surface. When there is an antigen coming, it will recognize that particular B cell that will fit into that antigen. And then this B cell will be activated to make a lot more antibodies. So that's how antibodies are made. But then depend on what B cells are being activated, sometimes they see this part of the antigen and sometimes they see that part of antigen. So you end up with antigen-antibody reaction. So in our body, when we react to an antigen, a foreign material, we usually make so-called polyclonal antibodies. That means that, so this is the antigen as A. Some B cell may see this part, and some B cell will see that part as antigen. And these B cells, like I say, are activated, and then all kinds of antibody are made. So we have usually a different kind of antibody that recognizing the same antigen. Okay, so this is our usual normal reaction to a foreign bacteria or viruses. It's polyclonal. So for the treatment purpose, polyclonal antibodies are too complex mixture. So then there's this invention of so-called monoclonal antibody. So how is that done? You start by immunize mice, okay, with the antigen of interest. And then the B cells in the spleen of the mouse will proliferate. Then somebody will remove the mouse spleen cells that's now containing a lot of these B cells that's making all kinds of polyclonal antibodies. So now you want to isolate these B cells. Now, our B cells do not live very long. When you remove them outside of the body, they die in a few days. They won't be immortal. So if you want certain antibody keep on being produced, so you have to make those B cells immortal. How do we do that? We fuse them with immortal cancer B lymphocyte. So usually this is called myeloma cells, okay? So by fusing the normal B cell with these cancer cell lines, these B cell fused cells are now immortal. So then you can select the kind of B cells or the kind of antibodies that you want. Usually you select for the one that has the strongest binding. So in that, this is exactly what was done in neuroblastoma anti-GD2. At the beginning, Dr. Reisfeld group, they basically injected mice with neuroblastoma cells and then fused those spring B cells with the cancer cell line, then select for things that will bind or recognize neuroblastoma but not normal cells. And that's how it came about, okay? At the time of selection, he had no idea it's GD2. We have no idea. We were just working together and came up with this anti-GD2, okay? So, with that in the background, then I will tell you a little bit about the current anti-GD2. I call them first generation. You heard a lot about them already. Then I talk about the newer generation of anti-GD2, and then some even newer ones. Some of them are covered yesterday, so I would not go into details. But I think I must share with you this slide. You heard about GD2. What is GD2? GD2 is 5-sugar with 2-sialic acid here, and it has a backbone of lipid, a ceramide. So it's a glycolipid, okay, sugar part and the lipid part. This is different from any protein antigens. All the anti-cancer antibodies on the market currently are targeting protein. This is the only one, or the first one, we hope, that's targeting non-protein. It's a sugar lipid molecule. So that's the initial study using Ralph's antibody. We showed that there are lots of staining. This brown color means it's positive staining. Blue color means it's control. I mean, this part, a stroma cell, no tumor cell, there is no staining. But you notice this is a blood vessel filled with tumor cells. So tumor cells here is positive, and tumor cell here, they are positives. So this also already tells us that it does not react very much with normal cells, but it reacts very uniformly with neuroblastoma. That makes this antibody a very good target. Okay, but as it turns out, it's not 100% specific. We later learned that it also reacts to skin melanocytes, peripheral nerve, and then weakly expressed in brain tissue. We also learned that it's expressed in other type of cancer, such as melanoma, glioma, small cell lung cancer, and certain sarcoma. And our first publication on this, I don't want to say how long ago, 1984, 30 years ago, okay? And a year later, Sloan Kettering, Nikon Chong's group came up with the 3F8. So that was, some of you may not have been born yet. But anyway, you heard about 3F8, so we'll skip that. So the initial antibody made is called 14G2A. That's a mouse molecule, totally mouse. And then later on, Steve Gillis took this V region. This is the determinant of antigen specificity. Which antigen to bind is determined by this FAB portion, okay? So he took the gene that dictating this part and combined it with the human IgG1. And that is a mouse and human chimeric antibody. That's why it's called CH1418 chimeric, okay? So I'm not going to go into the development history. It took a long, long time. But I just want to point out that the antibody, the mouse antibody, we filed IND 1988. A year later, we filed IND for chimeric antibody. And that is the very first antibody, chimeric antibody filed to FDA. As you heard a lot about Herceptin for breast cancer, rituximab for lymphoma, those were filed 1992, 3 years later. So I had the fortune of working with FDA officers who said, we don't know what are the regulation and the rules to approve a chimeric antibody because it's the first one that's made of recombinant DNA technology. So we went back and forth, what was done to this kind of cells. So it does involve viruses at that time, okay? I mean, it does even now. So with that, you have to worry about the virus contamination. So we went back and forth for 2 years. All kinds of tests are being done, you know. Finally, it got approved in 1991. That's when I was able to conduct the first chimeric antibody trial. So these are all studies done at single institute. After the initial success in those relapsed or refractory patients, showing that it has tolerable toxicity or controllable toxicity, but clinical responses were seen, then we begin to do a combination with GMCSF. And the idea is that you want to boost the killer cells, and the killer cells, G and M, are both killer cells. So granulocyte, monocyte, colony stimulating factor were used to induce more of them and also to boost their killing activity. So that was also confirmed by the POG study in 1994. So by then, with the confirmation, logically, you want to go to the randomized phase 3 trial. That's what you all discussed about this morning. But at this time, we ran into a snag. By then, we have been through 3 biotech companies. The very first one gave us the mouse antibody, and the Damon is the one that produced the chimeric antibody, and Repligen then licensed the Damon, which is a mid-sized company, and they made the supply for the POG study. Then they also licensed it out to a biggest company, I thought, Abbott. But Abbott, even though it's a big company, they like some other product, but they decided that you know, this is such an orphan disease, no market, or small market. So they decided to shelve it. And that would be the dead end, okay? But at that time, Ralph told me to go to Washington, D.C., because NCI has this Decision Network Committee. It has changed name. It's still there. It's called NEXT, New Experimental Drug, something like that, okay? But anyway, when there is sufficient evidence to suggest efficacy, but no commercial partner or no industrial sponsor, then they would review the data you have and then make a decision whether they will use government resources to make it. So at that time, I had to take literally in my suitcases all those CT scans. This is before the era of the disc, okay? So you have to do the X-ray films and the bone marrow slide and flew to DC and to have an independent panel review all the evidence. And luckily, they were convinced that there are enough activities, so they commissioned this biopharmaceutical resource branch at NCI. It's in the military base, by the way. I can't remember the name, Fort Detrick, that's right. So I had to make a visit there too, to tell them why you are making this for us. But anyway, it took a while for the government to produce enough batch and test it okay to go to phase 3 study. And this is the study that you now know very well. You know, it's a randomized phase 3 studies. The design is that we don't change anything in the frontline therapy, and if the patient reach at least a PR, a partial remission before stem cell transplant, then after transplant they receive radiation, then randomized to standard of care, which is cis-retinoic acid or cis-retinoic acid plus immunotherapy. So our goal was to randomize 386 patients, hoping to see a 15% difference. And this is also, familiar to you. Course 135 is for GMCSF, and course 2 and 4 is IL-2. And this is based on early data from Paul Sandel's group showing that 14G2A and IL-2 in mouse model is better than just 14G2A alone. So he also did a clinical trial, although we never knew whether cytokine is important or not, but it was done that way. So it came that way. This slide you've seen a lot of times, so I'm going to skip over. So this is the 226 patient randomization. We didn't reach the 386 yet, but then we had this conference call to talk about what to do with this data. Since it looks like p-value is highly significant and the improvement of immunotherapy is significant for event-free survival, for overall survival. So the decision was made to stop randomization. It felt that it's good enough and p-value is good. So from then on, the study was closed for randomization, but everybody who's eligible will be assigned to immunotherapy to collect more data. Now, there are 25 patients initially in that first 251 patients. 25 of them did not get randomization because they have biopsy-proven residual active disease. So ethically, we don't think they should be randomized. And even in those settings, we see that 1/3 of them, 32%, are event-free at this point. And the 53% have overall survival. So what did we learn? So the randomization was stopped, and right now we have continued to accrue patients. As of yesterday, we have 1,300 patients enrolled. And after the randomization was open, the data was open, FDA asked us to collect the so-called comprehensive safety data. In the phase 3 setting, in COG, we don't usually collect grade 1, grade 2. But to get regular approval, they want to do everything, including grade 1 and grade 2, a little bit of headache, a little bit of something it has to be reported. So we did that with 105 patients. They also requested that we do EKG monitoring with tracer. So you probably all know most of your children getting this antibody will have EKG monitor, but we never took the record, so we have to do that, and that was also completed. As of January this year, the company that licensed this product is called United Therapeutics. They made enough supplies, so as of January of this year, we switched the supply from NCI to UTC. So it's being, now all the patients are receiving the UTC-produced antibody. We submitted the BLA to US FDA in April of this year, but I understand that it was also submitted to the EU, the EMEA, about the same time. They have come back with some more questions, and then responses were given, but we are still waiting for final reply. Hopefully the latest will be next year, they should tell us. So we did have a price to pay, okay? Even though there are some, evidence of efficacy, but it does cause significant side effect. Neuropathic pain is the worst. And the vascular leak syndrome, which is usually associated with IL-2 courses, allergic reaction, skin rash, fever, and the heart rate increase, and the blood pressure changes, and so forth. But I guess fortunately, these are controllable and reversible side effects. And from early on, we know these side effects are rate and dose dependent. So below certain dose threshold, there's no toxicity. 10 milligrams per meter square is sort of the cutoff. So where do we go from here? The first question is, are cytokines necessary? We didn't answer that question, actually, because it was given as a package. GMCSF, IL-2, as a package. We didn't know whether it's necessary. But cytokine seems to helped to answer the IL-2, at least the way it's given with subcutaneous IL-2, it did not make a difference. So maybe IL-2 can be done away with. We don't know that for sure. And then does the GMCSF help? Again, we don't know that. And there's no randomized study done. But based on what Shakil told us yesterday, there are sequential study of 3F8 without GMCSF, and then sequential studies show that when GMCSF is given in the most recent patients, the survival is better. But we don't know it's because of the improvement in care or is that really, really matters. Okay. And then the next question is, can we use it for salvage treatment in the relapse for second remission? At least in Sloan Kettering, using 3F8 and GMCSF, 81 patients in second remission is now long-term. 16 out of 81. So about 20% of them are disease-free now. And the next question will be, how do we improve the efficacy or reduce the toxicities? Shall we increase the number of courses? Not at this moment, it's really quite toxic. So perhaps Dr. Lode yesterday told us about less reduced toxicity with continuous low-dose infusion. Whether that would give you the same efficacy or not remain to be proven, but at least it has less toxicity. And we heard from Kate Matei yesterday. You can use something called lenalidomide to mimic IL-2 and GM-CSF. This is a drug already approved on the market for adult, and it does increase NK cells, which is a form of killer cells, ADCC, and cytokines. Actually, it increase IL-2 and GM-CSF. But the beauty of it, it doesn't have that cytokine-induced toxicities. It's fairly well tolerated. So it's ongoing. What about chemotherapy plus chimeric antibody? That is certainly something to consider. I will show a slide later on. And then another option will be to use with the very hot molecule, so-called immune checkpoint. If we can block immune checkpoint, perhaps the anti-GD2 efficacy will be better. And then another option will be to combine it with the NKT stimulatory glycolipid. I'll go down the road with you together. So this is the adult experience showing that when you have the rituximab, anti-CD20, when it's given together with chemotherapy called CHOP, there is a synergism. The survival is much better. And the same thing is true for breast cancer. FEC is a chemotherapy, and when the green line shows the combination of FEC with Herceptin, the antibody. So with that, COG has already started a phase 2 trial for relapsed refractory neuroblastoma, and combining irinotecan, temodor with chimeric antibodies. So we will wait to see what is the result. Now I mentioned briefly about the hot molecules called immune checkpoint. These are molecules that, let me go back. Okay, so when the antigen-presenting cells present the antigen to our T cell receptor, the T cell is activated. At the same time, there is a molecule called CTLA4, which is normally present inside the T cell. It will now, during the process of activation, it will now come out to the surface, this orange bar here. And this acts like a brake, the car brake, to stop the T cell from perpetual activation. Otherwise, it can cause autoimmunity or something. Too much is not necessarily good. As it turns out, in the cancer tumor microenvironment, many times the T cells are paralyzed because CTLA4 is in excess amount. So the T cell cannot act. And the same thing, there are other molecules such as PD-1. So this is another checkpoint molecule. So the PD-1 is another block that will stop the T cell from activation. And many tumor cells actually express its ligand, so its binding partner, called the PD-L1. Now we already know in 2011, CTLA4 antibody made a big difference in the survival of stage 4 melanoma. Okay, so it's approved, and only this year, July 4th, anti-PD-1 was first approved in Japan for melanoma also. And now in September, it's approved in USA. So this is a very hotly pursued area, and we think that if we can inhibit this pathway, the immune system will be activated. So they are, oh yeah, I guess I'm ahead myself. So this is anti-CTLA4 for melanoma and anti-PD-1. Now they are beginning to do combination study. So the rituximab with anti-PD-1 actually in relapsed lymphoma. It looks promising in the phase 2 study. Herceptin and anti-PD-1 in animal model also look very promising. So in my lab, we have been looking at the chimeric antibody with anti-PD-1, and that looks good too. But then we need to convince some pharmaceutical company to give us anti-PD-1. So now I want to switch to our second generation quickly. I think some of them you already heard. One of them is so-called immunocytokine. It's a fusion molecule, and I'll just go straight to it. So this is a humanized anti-GD2 combined or conjugated to 2 molecules of IL-2 right here, okay? So the idea is that T cell or activated NK cell will have receptor for the IL-2. And the anti-GD2 will be attached to the tumor and then will attract the T cell or NK cells to the site and then do the killing. So basically, phase 1, phase 2 study have been conducted, and the maximum tolerated dose is smaller than the naked antibody because of IL-2. You cannot give too much IL-2. And we have done even a simulation of phase 3 study. So the fusion protein is given together with the GMCSF and cis-retinoic acid. And this study is completed, result is to be published. We haven't heard about it yet. And then you heard about humanized 3F8. There's also a humanized 1418 by Steve Gillis. But he did a little modification. He changed one amino acid at the 322 position from K to A. Why did he do that? He wanted to reduce the pain. Okay, so we know in our early studies in the rat, rat is a good model for pain. If you use complement deficient rat, the anti-GD2 does not produce pain. So we know it's a complement dependent process. So based on that, he made this change because that is a site that's important for binding to the complement. So as expected, we showed that the humanized antibody, this mutant antibody, has very little CDC, complement killing. But the ADCC, killer cell killing, is still pretty good. So, and this is my collaborator, Linda Sorkin, who used the rat model. Just look at the y-axis, which is the bar for the pain level, okay? So this is the chimeric antibody, and this is the mutant antibody. The pain score is much less when we use this humanized antibody. So a phase 1 study has been completed, and indeed, because of less neuropathic pain, they were able to push those way up to 70 milligram a day per meter square. Right now, we are using 25, okay, in COG, and in Cyopen it's 20. And with Holger Loden it's 10, Okay, so this is really significantly bigger dose. Whether the bigger dose is useful or will be more efficacious, we don't know yet. Now there is a new antibody. I think John mentioned briefly in his last talk. He said the antibody ideally he would like to use is a new antibody against O-acetyl-GD2. It's just a relative of GD2 with a little modification with an acetyl group. It's also highly expressed in neuroblastoma, like in the middle one. Okay, it's dying out. Anyway, the middle panel, the brown color, is a neuroblastoma, and this side on the right side is a nerve fiber. As you can see, it has no staining in the middle panel, but at the A3, which is neuroblastoma staining with our current, CH1418 or 14G2A, there is reaction to the nerve fiber, okay, and that explains the pain that our children is suffering from. So hopefully, with this antibody, it will have less side effect. And we did a study together again with a rat. So this is the lower panel showing again the chimeric antibody has high level of pain score, and the lower with this new antibody against O-acetyl-GD2. But in terms of anti-tumor activity, it's really comparable. So I think this is a promising next generation anti-GD2. Okay. Now I want to switch gear to anti-IT. It's a vaccine. So you heard about the Holger talking about the anti-IT. This is similar approach to the same principle. So this antibody. So if we look at this tumor, the circle here is our GD2. So AB1 is CH1418 that we are now treating patient. But you can also take this antibody to treat a mouse instead of a patient. And then look for AB2, something that will react to this anti-GD2, so that AB2 is so-called anti-GD2. Hopefully, this you select for the AB2 that will block the binding or compete with GD2 binding. To do that, the AB2 would have to look like GD2. So basically, you select for an AB2 that looks like or pretending to be a GD2. Then you can use this molecule to be a vaccine. So why bother? Why not just to use GD2? You heard about GD2 is a good vaccine. But GD2, remember, it's a sugar lipid molecule. So the immunogenicity is poor. And you have to use a lot of adjuvant. That's not the point. But majority of the antibody made in GD2 vaccine is IgM. And what you really want is IgG type of production. Okay, but so we were hoping that this AB2, we call it 187, would mimic GD2 as a vaccine. Toxicity-wise, it's wonderful. It's really has, we didn't see any of those significant toxicity you see with chimeric 1418. Majority, the major toxicity is this site of injection. You inject like a vaccine, So this is the area of inflammation. But it goes away in 3-4 days. There's no neuropathic pain you see. And then, most importantly, does it really induce anti-GD2? So does it make patient make their own anti-GD2? And the answer is yes. They are actually, it takes a while though. It takes about 6 weeks to begin to see this endogenous anti-GD2. But then if you continue to immunize them, about 6 months it reached the plateau and it will stay there for quite a long time. And are the antibody functional? Can they really kill tumor cells? And the answer is yes. that if you just take patient serum after immunization, they are able to kill neuroblastoma cell by complement or by ADCC mechanisms. And as I mentioned, toxicity is not seen, except for the local reaction. And what about outcome? This is not a randomized studies, and they are patient selection bias, I must say. So 16 of 21 patients enrolled in first remission remain to be disease-free. But if you come in as a second remission, 1 out of 10 is still disease-free. So we really need randomized trial to show if the vaccine is really active. In adult, they did a study on melanoma, the same thing. They show that it is, not toxic, and the patient can make anti-GD2. But again, we stopped there because the company went out of business and we're stuck with it now. Okay. And then the third generation, I think Shaquille told us about the DDKLH vaccine, and I know UK is going to participate in this trial. And we also heard about the TCAR, so I'm going to skip this next set of slides. This is my last slide that summarizes all the players that I know of, okay, on this slide. So 3F8, we already know, the status is phase 2, effective, but it has toxicities. It also induced HAMA, human anti-mouse antibody. And humanized 3F8, that's in phase 1 right now, is supposed to be less immunogenic. And then the chimeric antibody, 4 doses, or 5 doses, depending on Europe or USA, plus cytokine, phase 3 studies, effective but toxic, and they are late relapses. What about low-dose continuous infusion? Phase 3 studies will be ongoing in SIOPEN. The efficacy, hopefully, will be similar, but we don't know that. And the fusion protein, the humanized antibody with IL-2, it's in phase 2 stage. I guess the drawback is that you cannot give as much as you like, so lower dosage. And the toxicity is similar. Really, it's not better. And the human mutant, humanized antibody, phase 1 study is done. It does allow higher dose because of less toxicities. And then the anti-AIDS vaccine, I just did the pilot study. It is immunogenic, and it has little toxicity. We don't know about its efficacy yet. And then the anti-O-acetyl-GD2, this is preclinical. We expect it to have little toxicity, but it's still too early to know. We need to start from phase 1. GD2 CAR, we heard about it. And the GD2 vaccine, as I mentioned, it's phase 1, immunogenic, little toxicity, but mainly IgM production, okay? And then chimeric antibody with either compound 34 or anti-PD-1 is still early in preclinical phase. We hope it can improve efficacy and with reduced toxicity. I think I will stop here, and thank you very much.